So we're doing quiz time now. We just finished talking about the pancreas, and we're going to finish up with the large intestine in a few minutes, and then just go back and review all the different types of digestion. So what releases digestive enzymes into the mouth? And so if you think about the mouth, the liquid that's in your mouth is called saliva or spit, and the thing that makes saliva are salivary glands. What releases digestive enzymes into the small intestines? So we just said salivary glands make saliva into the mouth, so it would not be that. And then this next part is tricky. All three of these things release something into the small intestine. The difference is that this stuff secretes bile, which is actually not an enzyme. So the pancreas is the one that secretes digestive enzymes. And the pancreas secretes a ton of enzymes, enzymes that break down carbs, and different ones that break down lipids, and different ones for proteins, and different ones for nucleic acids. What releases bile into the small intestine? So I just gave that one away. That's the liver and the gallbladder. Here's the large intestine, and this is our last organ to go over. So this is the very end. Food starts out in your mouth, goes to your pharynx or your throat, down your esophagus to your stomach to your small intestine. In the small intestine, digestion is completely finished. So whatever makes it into this large intestine is poop, and it's liquidy here and it's solid here. And so the major thing that the large intestine does is it absorbs water from the intestines into the blood. The other thing that it does, there's a lot of bacteria in here, and hopefully they're good bacteria, and they actually make vitamins for you. So vitamins, water, and ions are absorbed into the blood from the large intestine. And again, it's not large because it's long. It's really not particularly long, but it's large because it's wider. The diameter is big. So its major function is to form and store poop, otherwise known as feces. So here we're going to summarize all the different types of digestion. So we'll start with carbohydrate digestion, and we'll talk about lipid digestion, and protein digestion, and nucleic acid digestion. So these are all great carbs. Polysaccharides are the polymer of carbohydrates, so we'll start with them. Polysaccharides are broken down into disaccharides by an enzyme in the mouth. It's called salivary amylase, made in the salivary glands, but it's used in the mouth because food doesn't actually make it into your glands. And then later by the same enzyme, this time made by the pancreas, so later by pancreatic amylase. And again, food doesn't actually make it into your pancreas. Food is digested in the small intestine with enzymes that got secreted from the pancreas. And so once you break the polysaccharides down into these disaccharides, you just have to break this bond to make the monosaccharides. So disaccharides are broken into monosaccharides by enzymes stuck in the small intestine wall. And here are some examples: maltase, sucrase, lactase. They break disaccharides down into monosaccharides. So for example, lactase is an enzyme that breaks down lactose, which is the milk sugar, and it breaks it down into the monosaccharides, I think galactose, and the other one, is it fructose or glucose? I think it's fructose, so I might be wrong with that one. But the idea is you have a disaccharide here, and you're breaking it into a monosaccharide and another monosaccharide. So for those of you who are lactose intolerant, it might be because you're lacking the enzyme that breaks this bond, the enzyme lactase. And I'm going to skip the video because they don't screen capture, so I'll show you that separately. So protein digestion, this is a big protein polymer, which is called a polypeptide. And so what you're doing here is breaking a polypeptide into smaller polypeptides, or into peptides. So pepsin is an enzyme that's in the stomach, and it breaks down proteins to smaller peptides. Instead of a polypeptide, it would still be a polypeptide, but it would be a smaller one. And then the next step is pancreatic enzymes. I'm just going to call them a general term proteases. So that means protein, and then ase, an enzyme that breaks down proteins. And this is in the small intestine because food does not actually get into your pancreas. That's supposed to be underlined. Your pancreas just makes the enzymes. And so it breaks these proteins down again into even smaller peptides. So here's a small peptide being broken into an even smaller peptide. And so if you look at this, this is a pretty small peptide. There's only 1, 2, 3, 4 repeating units. And so what you're doing now is breaking these things into their monomers. And I wish the monomers were called monopeptides, but they're not. These things are called amino, M for mono, amino acids. And so this whole nitrogen, carbon, carbon with an O, nitrogen, carbon, carbon with an O, nitrogen, carbon, carbon with an O. That whole repeating unit is now down here, nitrogen, carbon, carbon with an O. So this is an amino acid, and this is a different amino acid. And they're different because of this thing called the R group. You can see this amino acid over here doesn't have much of an R group. It's just got a hydrogen sticking off of it. So these are enzymes that are found in the small intestine. So this is a peptide. These enzymes are called peptidases or peptidases. And they're on the small intestine wall. And the main thing is that you're breaking peptides all the way down to amino acids. They can be absorbed into your blood and go to all the different cells of your body and be used wherever you need to build proteins. Lipid digestion. The first thing you start with is not actually an enzyme. Bile salts or bile is something that physically breaks down lipids to increase their surface area. So if you look at a glob of fat, let's say even this glob, it's already been broken down a lot compared to that one. Bile will break it down even further. So if you consider the surface area of this, now what you've done is given it an increased surface area. So before it just had this outer surface. Now it's got all this space for the enzyme to come in and say, ooh, let's go here, let's go here. And then you have your enzymes from the pancreas. So this is lipids that we're breaking down. So the enzyme is lipid ACE, so lipase. Pancreatic lipase, so enzymes that are made here, secreted right into the small intestine, break lipids into what they're made out of, fatty acids and glycerol. And of course it happens here in the small intestine. Please keep pausing this to write down stuff and rewinding it if you need to. Here's another picture of it. This is one of the things that you built during our lab when we built molecules. This thing is glycerol, and the things hanging off are the fatty acids. So if you break these off, you're going to get glycerol and 3 fatty acids. And that's just what lipase does. It breaks down lipids. The last one is nucleic acid digestion. And you probably guessed it again, a lot of the enzymes come from this pancreas, and they're secreted right out here to the small intestine. So pancreatic, let's see, what should we do to name this? Nuclei, and then put an ACE on it. Nucleic acids are broken down by nucleases. ASE. So pancreatic nucleases, remember they always end in ACE, ASE. In the small intestine lumen, break down nucleic acids into their monomers, actually, which are called nucleotides. But the thing is, so here's what nuclease does. It will break a bond here and here and here and here. And so you're going to get all these nucleotides. Now the next step that has to happen is there's another enzyme that will break down these nucleotides by breaking off the sugar and the phosphate and the base. And so enzymes in the small intestine wall break nucleotides down into what they're made out of: sugar, phosphate, and bases. And so pause this to make sure you can write all that in. And here we go. Here's the sugar, either ribose or deoxyribose. The base is either A, T, C, or G, or if it's RNA, it could be U instead of T. Those two, one's for DNA, one's for RNA. And then the phosphate. Whoops. So I'm breaking the bonds there. Okay, so here's a summarizer. You need to go back to page 2 to do this one. And so you already have polysaccharide broken into disaccharides, which are broken into monosaccharides. That should already be part of your notes. So now what I want you to add in is right here, what are the enzymes that break these things down? So the enzyme that breaks down polysaccharides actually starts in the mouth. It's amylase. So salivary amylase in the mouth. And perhaps you remember that there's another enzyme also called the same thing, but it happens, it's made in the pancreas, and so that's pancreatic amylase. And it's in the small intestine lumen. Lumen, I don't know that I defined. This is just the space. It's just the hole or the empty spot that's inside the intestine. It's the space inside the intestine. So pause this and fill that in. Disaccharides are broken down into monosaccharides by enzymes, specific enzymes. So sucrase will break down the disaccharide sucrose. Maltase will break down the disaccharide maltose. Lactase will break down the enzyme. Will break down the sugar or the disaccharide lactose. So OS is for the sugar, ACE is for the enzyme, and that happens in the small intestine. Lipid digestion, we're breaking down, we'll talk about triglycerides for now because that's your main, that's the main component of your diet as far as lipids go. So you're breaking triglycerides into fatty acids and glycerol. So you should already have this in your notes. I think it's on page 3. And so the thing that breaks it down is an enzyme called lipase. So pancreatic lipase in the small intestine lumen. Make sure you're pausing this so you can go back and write it all in. Protein digestion. Again, I think page 3 of your notes. So you've gone backwards to do this, hopefully. So polypeptides are broken down into smaller peptides, which are broken into amino acids. And so now you have to do the enzymes. The first one is pepsin in the stomach. So there's pepsin in the stomach. And then pancreatic proteases. So this is protein with ACE on it in the small intestine. Again, we're doing all the enzymes that are secreted by glands go into a tube like the small intestine or into the mouth. And then peptides get broken down by peptidases. So peptidases in the small intestine wall break it all the way down to amino acids. So the big picture that I want you to get is that polypeptides, proteins, are broken all the way down to their monomers. This should be the monomer right here. The monomers are amino acids in this case. And then the one other interesting thing about proteins that's different from the other nutrients is that your body's made mainly of protein and you don't want to digest your body. So each of these enzymes is actually secreted in an inactive form. And that's good because you're not going to digest yourself if they're made in an inactive form. Once they're secreted into the stomach or the small intestine, they're activated, but it's okay because if this is your intestine, you've got a layer of mucus to protect you. Last one, nucleic acids. So you should have this. It's on page 4, I think, of your notes. And so you should already have this part filled in. Nucleic acids are broken down into their monomers, nucleotides. And then these are complicated monomers. They're actually broken into the parts that they're made out of. Sugar, either ribose or deoxyribose, phosphate, and the base, either the A, the T, the C, the G, or the U. And so if you're going to name an enzyme that breaks down nucleic acids, you might call it a nuclease. And our favorite enzyme secreting organ is the pancreas. So pancreatic nuclease, and of course it goes into the small intestine. Nucleotidase, if you're going to break down a nucleotide, that's the little one, you would name it nucleotidase in the small intestine wall. And that is just about the end of our story. Let's do quiz time. Enzyme names usually end with what suffix? So I'm going to say ase. Os is for sugars, and I made those two up. Most enzymes are, you have to know this one, almost all of them are proteins. What do proteases break down? Well, prote sounds like protein, so they break down proteins. Why are proteases released in an inactive form from the organ that made them? And so the reason is this first one, so they don't digest the organ. They can be active whether there's protein-containing foods or not, although they could be wasted. But anyway, so they are only used when there's a lot of food in the stomach. Whatever food's in the stomach you want to digest. So they can help to produce mucus. Nope, there are different cells that produce mucus for you. What changes pepsin from its inactive form to its active form? This is not a big deal, but this is the acid, hydrochloric acid in your stomach. Certainly wouldn't be ultraviolet light because the light can't get all the way into your stomach. The yummy smell of food, you actually secrete saliva because of that. But okay, so what do lipases break down? Let's see, lipases. How about lipids? This is the only really hard one, I think, amylase. None of these make sense, but the answer here is that amylase breaks down carbohydrates. You might also get the same exact question, and carbohydrates won't be there, but polysaccharides will be. Could get really tricky, and it could give you a polysaccharide example like starch. Oh my goodness, then you have to think through like 10 things for that one. What do nucleases break down? They break down nucleic acids. And why isn't bile an enzyme? because it doesn't chemically break down anything. It actually just physically breaks stuff down. Besides making bile, what other important thing does the liver do? It detoxifies your blood, which we haven't talked about too much yet, but that's important. All right, that's the end. Bye.